DNA molecules are double helix-shaped molecules in which nucleobases are attached to a sugar-phosphate backbone. The order of the nucleobases encodes the amino acid sequence of proteins. Thus, DNA determines the characteristics of living organisms. In the sugar-phosphate backbone, deoxyribose and phosphate alternate. The nucleobases are classified into two types, purines and pyrimidines. Adenine and guanine are purines. Thymine and cytosine are pyrimidines. According to the rule of base pairing, there must be a thymine opposite an adenine and a cytosine opposite a guanine. That is, a larger purine is always paired with a smaller pyrimidine. Nucleobases are attached by hydrogen bonds. Adenine is bound to thymine by 2 hydrogen bonds, and guanine is bound to cytosine by 3. Eukaryotic cells with nuclei measuring only a few micrometers may contain several meters of DNA, which has to be packaged and organized in order to fit into the tiny nucleus without tangling, and to ensure that it can be divided appropriately during cell division. The DNA molecule is wrapped onto clusters of histone protein molecules to create nucleosomes. 8 histone molecules make up the core, with the DNA coiled around it twice. DNA is attached to the core by H1 histones. These histone cores form a structure similar to beads on a string. This beads on a string structure coils into a spiral-like solenoid structure with 6 nucleosomes in one section. In the next stage of packaging, parts of the coil attach to a protein skeleton and form a characteristic loop structure. The loop structure coils again to form the chromosome. Human cells have 46 of these, with a total of nearly 2 meters of DNA. That is, each of the microscopic chromosomes contains 4 centimeters of DNA on average.